Catherine.
Oh no, these are the new people.
Yeah, just new people. If you're registered, don't worry. Okay, who's raising their hand?
Did you say a no-go?
Yeah. Okay, so let's get going here. Okay, so today we're still in molecular biology. That's the big news. Okay, so some quick business. All people trying to add this class, I just took your attendance. Make sure you're here. If you're not, you're gone. So a few people are crossed off the list. You need to let me know at the end of class, meet me outside again, what if you have a requirement for a recitation a certain day, because I would like to just put you wherever you fit. But if you have an absolute requirement, come see me at the end of class. And I'll let you know by 5 p.m. today if you're in. And then you can just pick up an add drop slip from my office. Okay. Recitation next week. No one can ask me about that. I forgot to tell you guys that there's an oral exam option. You all know that because you all read my syllabus in its entirety. But I'll tell you anyways for those who didn't. Actually, I saw a number.
Why don't we just take some chairs in from MB3 into here, just put some tape or something on the back that says MB3, since you're going to need them long term.
Well, a lot of these people I'm going to kick out of the class if they don't behave. No, okay, yeah.
Yeah, you can test tomorrow, and if they fail, yeah.
Yeah. Okay, yes, definitely. I'll come by after. So there's an option for oral exam for each. So everyone has a chance to replace one exam with an oral exam. Now, it doesn't mean take the exam, fail it, and then come to me and say you want an oral exam. It means that you just say to me, I'd like to try this next exam as an oral exam. So look in the syllabus, there's some rules about that. It's a good option for people who have huge amounts of test anxiety and feel like they can tell me about it, but they just can't get it out on paper during an exam when there's a time limit and all that stuff. The way it pretty much works is it'll be the same topics that are on the normal exam. I'll put it into a random generator. You'll come into my office. I'll hit randomize. Whatever comes up, I'll orally question you on for like 10 minutes. And I'll question you until you can't answer anymore. And so you will feel like you're doing badly because I'll push you until you say, I don't know. Shut up. No. I think they're testing. And so just keep that in mind. And the week before the exam, I'll say, hey, anyone who's wanting an oral exam, email me and I'll put up a schedule or something like that. Okay. Don't forget, pre-test is due 5 p.m. Friday. I'm seeing people are taking it, and people are doing very well, which is good. Don't forget, the weekly quiz begins on 5 p.m. on Friday and goes through Sunday, and it is every single weekend. So get that in your head. Every weekend there will be a quiz. Don't forget it, because that will hurt. Okay, there's the weekend. There's the business. So is everyone sitting relatively in their groups from the other day? Okay, you suck. Okay, so just sit. I'm not going to have you reshuffle because that's annoying. So just turn to the groups of you, like 3 or 4 people around you, and we're going to finish where we left off. I do want you to try to sit in those groups I put you in. I could send it out, or just remember where you were sitting. Who remembers where they were sitting? You can't see. Okay, we'll have to work that out. But these people can never see. Okay. All right, you sat there all last semester. Okay, well, you can stay there. You put your time in. Okay, so, turn to your friends, and we're gonna. And we're gonna go back to what we were doing because we had like 5 minutes to do it. So you've got your eukaryotic cell, right? You have a eukaryotic cell, and you drew it, and you drew your nucleus, right? We got that far, and we are talking about. I'm having one of those feelings like when I'm talking to my kids, and I'm like, get ready for bed, get ready for bed, get ready for bed, and they're like, blah 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 blah, and they're busy talking to each other, and I'm like, I said, get ready for bed. Anyways. You know, forget it. That's the feeling that all parents have when they feel like they're talking to people who aren't listening. Okay, so you've got your cell and you've drawn your organelles in. Okay, so start there, finish up drawing in your organelles and everything in it. I'll give you 2 minutes if you didn't finish it the other day, and then we're going to continue our conversation. Okay? Or do you not want to do this? And should I just tell you what you need to know for an exam? You guys suck. Okay. No, you're doing my activity. Gosh darn it. So finish drawing. Do you have all your organelles and are you ready? Okay. Then don't take 2 minutes. Okay, so we were talking about what happens in the nucleus, and then we were talking about what happens in the cytoplasm. So what was happening in the nucleus? We were talking about what was in there. What's the main job of the things that go in the nucleus? Transcription, DNA replication. Okay, good. And what's happening in the cytoplasm? Translation. What else? Metabolic processes. Yep.
Parts of signaling.
Signaling. Some parts of signaling. Right. Right. Yep.
Vesicular transport.
Vesicular transport. Yep. Glycolysis. Okay. So we're going to mostly focus on what's happening in the nucleus in this class, but today we're going to start with talking about the big picture because it's really, really easy to get bogged down in these teeny little details, like what is the structure of a nucleotide. And I don't want you guys to be getting bogged down in those details because that's not useful. If you can't think of the cell holistically as part of a larger thing called an organism, well, if you're not bacteria, you are the organism if you're bacteria, then it's really hard to think about gene expression. And to think about gene expression, you need to be thinking about it as something happening in response to something else, right? Gene expression is not just random. Okay, so let me turn off this light. Darker than the other day. So this is what I want you guys to remember, right? There's a person, and inside the person there are organs, which are made up of groups of cells of similar type, right? Kidneys are made of, these are actually kidney cells. I made sure of that. Kidneys are made up of kidney cells, right, which are different from what makes up brains, right? And then there's the individual cell. And what we're going to talk about in this class is what's happening in the individual cell, not only in the individual cell, but frequently inside the nucleus. Who's familiar with looking at immunofluorescence? People who've had me before are. I just want to kind of orient those of you who don't sit around looking at immunofluorescence pictures. So it's microscopy where you look at the cell and you take the cell and you stain them with different antibodies against different things so you can visualize different parts of the cell. So up here they're using propydium iodide to stain DNA. So every round circle is what? Every round orange circle is what, if that's DNA? A nucleus. So is that one cell or many cells? Okay, good. So that's a whole lot of cells, and each little round circle is a nucleus, so represents a cell. And then this is one cell. Here they're using DAPI to stain the DNA. So there's your nucleus, and they're using a red tagged antibody against actin and a green antibody, bless you, against mitochondria. So you can actually see kind of the edges of the cell, because that's the actin cytoskeleton, and then there's your DNA, and mitochondria tend to kind of cluster around the nucleus. So if I ever show these, then you can start to get used to seeing these kinds of images. So I just want you to keep in mind that even though we're talking about what's happening in one cell inside the nucleus, don't forget it's part of a whole organism, right? And I'm very eukaryotic centered or even mammalian centered. So you'll have to excuse me if I ignore bacteria. I tend to ignore bacteria a lot. Shut up. Okay. Whoever's listening to the recording is going to be like, what is she doing? Because probably didn't hear the alarm going off. And when I say you're part of an organism, it's because I want you to understand that gene expression is happening in response to the conditions around the cell, right? If you're a part of an organ and you're living in a cluster of cells all together, right, that makes up a kidney, let's say, you're going to be responding to things secreted by your neighboring cells. And those things are going to bind to receptors, and signaling is going to happen, and transcription factors are going to go into the nucleus and activate gene expression or turn off gene expression. And I want you to think big picture, okay? If you can start to think big picture, this will be an easy class. If you get stuck on the details, you will do really badly. Okay. Any question? Well, let me just drill in the big picture one more time. Right? So here you've got a cell in your nucleus, and you've got some sort of extracellular molecule that's being secreted. This is an example of immune response, where you've got cytokines that are binding to receptors on the cell surface, and they activate these signaling pathways. You need to memorize this for like a quiz in 5 minutes. I'm just kidding. God. Okay. This is big picture, guys. Don't memorize this. Cytokines binding to some kind of receptor, and then there's some kind of signaling, which means phosphorylation and proteins binding to other proteins, all to relay the message to the nucleus, which basically results in transcription factors going into the nucleus, binding to DNA, recruiting RNA polymerase to do what? What does RNA polymerase do? Transcription. That's making a copy of the gene that's going to eventually be translated into protein, right? So you get DNA, RNA, protein. So the whole point is, there's something going on out there. There's a virus, there's a bacteria, I don't know. Bad things are happening. Someone stabbed someone with a pencil. I don't know. And cytokines are being secreted because the cells in the area are going, whoa, bad things are happening. We need to multiply and get ready to fight the invader, right? And so then these cytokines are going to bind to the receptor. You're going to have signaling. Transcription factor goes in, binds to the DNA, and activates gene expression. With gene expression, you get a whole bunch of genes turned on, make a whole bunch of proteins. Maybe some of those proteins will be cell cycle proteins that will push the cell into cell cycle so that the cells can multiply and go out and fight the good fight. Okay? And so I just want you to keep in mind that this might seem like this big world you're stuck in, but it's like this is really small compared to like this large human that needs to survive. So, any questions about what I'm talking about? What do I mean by big picture? Everyone understands? Big picture. Okay. Okay. So, let's finish that drawing. So, what is happening in the nucleus and what needs to happen to make a functional protein in response to an outside signal? I want you to take 5 minutes to talk with your friends and think about what is happening in the nucleus. This is a very vague general question. You got to get used to them because I'm trying to get you not to think too specifically. Right? You've got what do you have in your nucleus, what needs to happen to get a functional protein to respond to an outside signal. So go chat with your friends, and I'm going to go out and get a bagel. No, I'm just kidding. And I'll check in a few minutes.
All right.
And like a more sticky more biology, you would like 28 papers. I'm the guy who put my sunglasses over.
Oh yeah, I saw you.
What mine though?
I know right now. I know it's
That's rare.
So confident. There's 46 minutes.
Right.
Generally speaking, we need some kind and then it has to bind to an activator and then activate calls already proliferate.
It depends on how like the code is. Like you can signal one thing at one time. They spell one.
Assuming everything is correct. And then the RNA, once it's synthesized, then you find the path to pollinate some
Are you confused? You're on it?
We won't multiply anymore.
Well, when you're replicating DNA,
Open-ended question.
Yeah.
What's happening?
How about that? Sure. Is there a question?
I put it Yeah, I'm taking this through to neurology and biology.
So many things are happening. That's the point of the question. Like all
Okay. Okay, guys. So who found that frustratingly vague? Okay, good. So the question was, what is happening in the nucleus and what needs to happen to make a functional protein in response to an outside signal? So I just gave you the example of the outside signal, right? There's some kind of fight going on in the organism and an immune response needs to be mounted, so cells are secreting molecules that are binding to receptors and you're having signaling. And then what? What needs to happen in the nucleus to end up with a functional protein? Yes? Okay, transcription factors need to bind to the DNA. What's the point of transcription factors binding to the DNA?
So it can set up so it can read the template of the sense strand.
Okay, so can read the template. So the template of what? Does it just bind anywhere in the genome? Are there certain places in the genome where transcription factors bind? What's their purpose? Yep.
Bind to the promoter to open up the promoter so polymerase can come in and bind.
They bind to the promoter, right. Okay, so promoter. What is a promoter? This is how an oral exam would go, by the way. Yeah.
Start of a gene.
The start of a gene. Okay, so transcription factors go into the nucleus, bind to the DNA at the start of the gene. Which gene? Any gene? The gene that's going to make the right protein, right? Did you ever wonder, there are all these proteins being made? How does a cell know which protein to make, right? Is it random? No, of course not. It's regulated by these signaling pathways that result in transcription factors to go into the nucleus. They bind to the promoter, the beginning of the gene, and recruit What do they recruit? RNA polymerase. To do what? What does RNA polymerase do? Transcribes, right, makes RNA. Okay, so you've got that. So we've got a transcription factor. It went into the nucleus, bound to the promoter. There are certain things that need to happen though before a transcription factor can bind to a promoter, right? Are you guys familiar with the fact that DNA exists in different forms, right? And sometimes it could be really tightly packed, and sometimes it could be really loose. Yes? Right, so you need the DNA to be open, right? Because if it's tightly wound around a histone, for example, will a transcription factor be able to bind? You have to think about this, here's your histone, and you've got your DNA wrapped around like this, and then you've got another histone, your DNA wrapped around like this, right? So transcription factor needs to come in and bind to the DNA. Is it going to be able to get into this teeny little space? No, right? So you need to change the architecture of the chromatin to expose the promoter and allow that transcription factor to come in, and then allow for recruitment of RNA polymerase and gene expression to happen. Okay, so now we've got RNA. Then what needs to happen? Yes?
It needs to mature.
RNA needs to mature. What does that mean? Does it mean it needs to turn 18? Are we mature at 18?
No.
I don't know. I'm not sure. Yeah.
Make it mature, introns going to be spliced out,
Right. So we need to remove introns. We need to cap. We need to. Oh no! Is it open? Weird. Okay.
You can't twist it, but you can pull it.
Oh. Interesting. So then I have to start just screaming at people who come in late. Okay. Capping, poly-A tail, right? And then what happens? So you've got your RNA spliced, and it's capped, and it's got its poly-A tail. Then what happens? Yes?
Transfers to an area where it can add to a ribosome subunit.
Okay, so where is that happening? Where does it associate with a ribosomal subunit? Yes?
Isn't that outside of the nucleus?
Right, so it needs to be exported from the nucleus, right? So what's really important for export of RNA from the nucleus?
Signaling.
Signaling?
Stability.
Yep.
Location sequence.
So a nuclear export signal, right? That's what. So there are these different. Yes, but that's not on the RNA, but it's going to associate with something that's going to export. That's going to export the RNA out of the cell. What's going to stop? Yep.
Hold on, what were you about to say?
What's going to stop? What's going to stop an RNA from going out of the nucleus? I'm just pushing you guys to see what you guys know. Don't worry, I'll teach it all to you. Kind of. What's going to stop? What if you don't have a poly-A tail? Are you going to get stuck in the nucleus? Maybe you can't go out. So part of export from the nucleus requires binding to different proteins that interact with your poly-A tail and your 5' cap. Okay. So if you don't have your cap, you don't have your poly-A tail, you don't have and if you do have introns, you'll never get out of the nucleus unless you're a viral protein that has ways of getting around things. Okay, so you're out of the nucleus. You're an RNA. You're properly spliced and capped and tailed and all those things. Now what? I heard something about a ribosome.
It attaches to a ribosomal subunit, and then the other subunit comes in and attaches to that, and then amino acids start coming.
Okay, so you've got association with ribosomal subunits, and then you have recruitment of the other subunits, and then you have translation. Okay, so now you've got a translated protein. Now what? Do we just hang out? Yep.
It gets packaged, I think, in forget what it gets packaged in, but it gets packaged and then it gets sent out.
Okay. Well, it depends on the job of the protein. So this is where we need to think big picture and realize there are many proteins that do many different things, and they're all going to go to different places depending on what their job is. So if you're a secreted protein, yes, you'll get translated into the Golgi, the ER, and then the Golgi, and then you'll get packaged into a vesicle, which is probably what you were thinking, and then you'll go on the secretory pathway and be secreted out of the cell. If you're a nuclear protein, then you're going to what kind of protein is a nuclear protein? Polymerases. There's a good example. Then you'll get translated on a free ribosome in the cytoplasm, and you'll get imported because you'll have a nuclear localization signal. You'll get imported into the nucleus, and you'll work there. Let's say you're a mitochondrial protein. You'll have a signal that's going to send you to mitochondria. And we cover all that in cell. But we're not going to talk about it now. But big picture, right? Because it's all connected. Okay, we've been doing good. Any questions about what we're talking about? Maybe? No? Okay. So we're essentially a bunch of clusters of cells, we as people. So how do all these cells work together to make a functioning human? Do they just all do their own thing? Yep.
They express different genes so they can each carry out individual tasks.
Right. They express different genes. They carry out different jobs. And what determines which genes are being expressed in different cells? Like compare a kidney cell to a liver cell to a neuron.
The transcription factors that are present and the wrapping of the chromatin.
Transcription factors, right? So different cells are going to have different levels of transcription factors. Which parts of the genome are exposed and available for gene expression. What else? But what all determines that, right? So transcription factors, heterochromatin versus euchromatin. Yep. Environment, absolutely, right? So big picture. The environment. If you are in a liver, you have a very different environment than if you are in a kidney or if you are in a muscle, right? Who took physiology of some sort, right? So you all had to learn this on a much larger scale, right? So the environment. And the cell can sense the environment how? Receptors. Receptors binding to what? Things, different signaling molecules, things secreted out of other cells, receptors on other cells. Okay, so let's have a quick example that we can discuss. So you're studying for your Bio 409 exam, and you're stress eating. Who stress eats? Who stress not eats? It depends. Yeah, sometimes I stop eating when I'm stressed. Sometimes I eat more. It's weird. It balances out, so I'm not obese. So you're stress eating. Your favorite food. What's your favorite food to stress eat? Salty, sweet, mix, chocolate-covered pretzels. Best of both worlds. Yep. Okay. So you're stress eating, and it's chocolate-covered pretzels, which means that your blood sugar is going to increase. So how's the body going to respond at a cellular and molecular level, right? Blood sugar goes up. I don't want specifics. What does the body need to do?
Get it back down.
Right. So the body needs to bring your blood sugar back down because if your blood sugar stays up, what's that called? Bad. It's called bad. Right? Diabetics usually have, when it's not controlled, there's all sorts of bad things that happen. Okay, so the body needs to respond, and then you've probably learned about like insulin and all that kind of stuff at some point somewhere. But what's all this doing? What's happening on the big picture level? So you've got high blood sugar. The body needs to bring it down. So where does that sugar go? Does it just disappear? It goes into the cells, right? So something needs to tell the cell we need to take all that sugar in from the blood, right? Yes, maybe, right? So I just want you guys to start thinking at this kind of macroscopic as you can get. Tell that to an ecologist. macroscopic level. So you can think about, okay, so what's going to cause, so you've got these what brings sugar into the cell? Does anyone know? Just guess. GLUT4. Okay, so that's specific. You can say some kind of transporter. Yep, some kind of sugar transporter. GLUT4. Yep. But how does the cell know to even make GLUT4? How does the cell know to even have it sitting at somewhere in the cell so it can go up to the plasma membrane and take in that sugar? Is it just magic? This is all gene expression, right? We've got insulin signaling, right? Insulin binds to receptor, gets signaling, gets gene expression. A whole bunch of genes are made, one of which is GLUT4, and it goes and it becomes its thing, and it goes and it sits up near the plasma membrane waiting until it needs to take in sugar. Well, it then takes in sugar. That's the very basic big picture. Okay, so well, we kind of just did that. Questions? So is everyone understanding the concept that gene expression is regulated, and it's regulated in response to the environment that the cell is experiencing, right? And so an example is high blood sugar, okay? You can have a million examples. Another example, an infection. Another example, I don't know, some toxins. I don't know. I'm sure we can come up with many. Who wants to give me another example of something that might induce a gene expression to occur? Injury, a foreign object? Yes, I guess so. How about hormones, different hormones? Cortisol levels, yep, hormones. Okay, stress that causes you to eat. What gene expression is behind stress eating? Okay, so we're going to switch gears now. Let's take a minute to think about our genome. Switch gears. Okay, tell me when you're done switching. Okay, so what is a genome? I know this is like totally 201, but we got to start somewhere. Huh? Who said that? So DNA, your DNA, that is your genome. So what does it contain? And I don't mean DNA, but so you've got all this DNA. What's the purpose of all of this DNA? What's in that genome? Like if you had to make a pie and split it into the different things that are in your genome, what might be there?
Mostly junk, isn't it?
It's mostly junk, or very important junk, depending on what you work on and how you're funded. Yes, mostly junk from those who aren't getting funded to study it, but it's very important if you study it. Yes? Introns, exons. Okay, so we've got introns, so mostly junk. So let's do like that. So we'll call this junk. And then here, what do we have? We have introns, exons, and what else? Coding regions, right. So those coding regions result in what? What do they make? Right, okay, protein coding, right. Coding regions. So these are going to make your proteins. And so you've got your introns and your exons. There's no actual proteins in your DNA. Just so you don't go and say, hey, Dr. Snodgrass, Dr. Ehrlich said there's protein in the DNA. There's not actually protein in the DNA. Introns, exons, what else? What other things are very important for encoding proteins or making proteins? What part of the genome is going to bind to transcription factors or RNA polymerase? Is that the introns or exons? Promoter. So these non-coding, there are these non-coding regions that are regulatory regions that are surrounding the gene, right? Okay. We're getting there. So all this junk, there are like these transposons and lines and signs and all these crazy things, which are really interesting. You should talk to. I had this fantastic conversation with Dr. Masters. Who's had Dr. Masters? He's great, isn't he? We love him. Oh, you work with him, right? And he works a little bit on this junk. So don't call it junk to him. He might be upset. Okay. So here's another question. So does genome size correlate with organism complexity? So if you have a huge genome, are you more complex than if you've got a teeny tiny genome? No. Okay, good. Good. So what do you think determines the complexity of an organism? Is it how many proteins you have? Yeah.
Your ability to alternate the splice.
Okay, well, so that's one thing. Alternative splicing, which does increase your number of proteins. Let's get a little bit more broad. How about your ability to, how you regulate your genome, right? Like, look at bacteria that not regulated nearly the way eukaryotic cells. Okay, so here's my figure to go with this, right? So somewhere up here, and like generally, these unicellular organisms do have smaller genomes, but and mammals are up here. But like, look at these salamanders and lungfish; they've got these gigantic genomes, and I would say are slightly less complex. Okay, right? So who's more complex? Who loves Monty Python? Does anyone? Okay, good. Who is more complex, right? So this lungfish here, who knows the band Lungfish? Oh, come on, people.
What?
The band Lungfish, anyone? No? Okay. Okay, who grew up in D.C.? Anyways, I won't judge. Okay. So lungfish, they have 132.8 billion base pairs, and humans only have 3.5 billion base pairs. So I would say that humans are a bit more complex, and I think they have a smaller genome, maybe, slightly. Okay, so it's not just about protein coding genes, right? No. Right, because this this bacteria here has 60,000 protein coding genes, and we only have 20 to 25,000 protein coding genes. So it's not about who has more protein coding genes. All right, so what's going on? Well, like we just said in our pie that we drew here, only about 1.5% of our genome is protein coding genes. And yes, most of it is junk. But it's pretty important junk if you think about how we're constantly assaulted. We're assaulted constantly with all these things to mutate our DNA, and this junk is probably pretty protective, right? If you think most of it's not protein. If our genome was only protein coding and we're constantly being assaulted with toxins and ultraviolet light and all these carcinogens and DNA damaging agents, would we have survived as a species? Probably not, because we'd be mutated like crazy and all our protein coding genes wouldn't work anymore, and that'd be bad. So I'd say the junk is good. Okay, so organism complexity is not due to genome size or number of protein coding genes, but to how genes are regulated and the number of functional proteins produced. Okay, so when I talk about how genes are regulated, what do I mean? Turning on and off genes, right? How whatever organism turns on and off their genes. Yep. And how many functional proteins are produced. Okay, so that brings me to this thought. One transcriptional unit, so one gene, can produce many different proteins. Those of you who've had me for cell, we've kind of walked through this a little bit. That can function in many ways. How? Right? One gene, many proteins, many functions. Right? So 500,000 plus proteins are produced from our 20,000 protein coding genes. That's more, right? 500,000 proteins are produced from 20,000 genes. Have you guys ever thought about this? And that's pretty ridiculous, right? So how does this happen? What are the mechanisms? Like I don't get it. So there's DNA and there's RNA and there's protein. How are there so many proteins and not that many RNAs? Or not that many genes, I guess. Does anyone want to take a stab at it? Yes. Okay, so processing, something about processing. Okay, so processing what? Structure of the protein. Okay, so we've got a lot going on here. So let's go back to processing. Processing. What is processed? That's gonna result in many different proteins coming from one RNA. Right. Alternate splicing. That's one. What else?
Post-translational modification.
So that's after. So if you have alternate splicing, you're going to end up with a whole bunch of proteins, and then those proteins can then be modified. Modified how? What is post-translational modification? Has anyone heard of it? Yeah.
Isn't that like adding on phosphorus, like phosphorylation and ubiquitination?
Yep. Phosphorylation, ubiquitination, simulation, methylation, acetylation, any ation, prenylation, glycosylation, Asians, all the Asians. Not Asians, Asians. Right, so we've got alternative splicing, making a lot of proteins, and then we've got post-translational modifications that are further expanding function. Okay, so I wrote something here. Right, so here's one mechanism, alternative splicing, so that you guys have learned about many times. So you've got your DNA, then you've got transcription, then your introns are spliced out, and you get your mRNA, which is made into protein. That's what you all know and love. And then sometimes, though, things change. You can get skipped exons, which result in a huge chunk being cut out. You can get just alternative splice sites. If you remove different parts of your RNA, you're going to end up with a different protein. Does that make sense? Right? Because how does RNA relate to protein? It's translated. But what does translation mean?
Taking the order of the codons and changing it into amino acids.
Codons, right, codons. So what's a codon?
3 base pairs.
Right, so codons, remember, 3 base pairs. You translate each triplet nucleotide sequence. Codon is translated into a certain amino acid, right? So if you cut out a huge chunk of your RNA, you will end up with a different protein because you're cutting out all these different codons, right? Does that make sense? Everyone gets that. Okay, so that was one mechanism. One that no one said is RNA editing, right? There are these different enzymes that edit RNA. There are cytidine deaminases that deaminate cytidine, so you'll get to these C-to-U, I guess not mutations, but changes, and that can put in an early stop codon and shorten a protein. An example is the apolipoprotein B gene, right? Apolipoprotein B. Does anyone have a guess on what that does? Lipoprotein. Sometimes you just need to read the word, right? Like everyone knows, like that a lipase is a what? An enzyme that breaks down lipids, right? So apolipoprotein B, you can probably guess it's some kind of protein that carries what? Lipids, thank you. And it exists in the liver and in the intestine in different forms. In the liver, it's the full-length RNA that's not edited. It's translated, and you get this 4,563 amino acid long protein. And in the intestine, it is edited. There's a cytidine deaminase that deaminates. So you get a CTU, which results in a stop codon right in the middle. This is not a mutation. This is on purpose. We want this. And it results in a protein that's about half the size. And one functions in transport of cholesterol in the blood, and the other functions in absorption of lipids in the intestine. So RNA editing, right there, you've got one gene, one RNA. It's edited just by one enzyme change, one base, two proteins. Okay? Okay, so we talked about post-translational modifications briefly, right? So these are post-translational. That means the modification happens on what? What kind of? It happens on proteins post-translation. After translation, right? So it's not going to happen on the DNA or RNA. And there are many post-translational modifications with many different functions, right? You can have just proteolytic processing. You can have a pro-protein that's cleaved to activate it, right? You see that in apoptosis with caspases. You can have post-translational modification dependent proteolysis, right? With ubiquitination, polyubiquitin chains are added to a protein that tags it for degradation. That's a way to turn things off. You can have post-translational modification dependent recognition, phosphorylation. How many times have you guys been told in a class, blah, 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 phosphorylation, blah, 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 blah? Like that. And that's how you remember it, probably. That's how I remember it until I got it. So when you phosphorylate something, you're changing its shape, right? You add a phosphate group that changes charge, that changes shape of a protein, and it creates binding sites for other things. So you'll get this phosphorylation-dependent recognition. You're creating a binding site so two proteins can interact and transport a signal somewhere or do something. Phosphorylation is just one example. Also, what we'll learn about with acetylation and methylation in terms of chromatin architecture and reorganization. That's very much based on these post-translational modifications of histones and how that affects histone structure or recruitment of other proteins and how that affects the DNA. And then you can also have these reversible multi-site post-translational modifications. That just means that reversible, you can add and remove. A lot of these things you'll get methylation and demethylation, acetylation and deacetylation. And that's how different processes in the cell are regulated, because with each modification, you're creating a new binding site for something to interact and some process to happen. Okay, so I created this very simplistic cell workflow, right? Big picture, right? So you get a signal from another cell, and that has an effect on gene expression, right? It has an effect on transcription. It'll either be positive or negative. If it's negative, then you'll get chromatin modification resulting in gene expression being turned off, no gene expression, tight packing of chromatin, no transcription factors will go anywhere, like all that will be off. If you have some kind of positive effect, you'll get chromatin remodeling, recruitment of RNA polymerase by transcription factors. Then you'll get transcription, processing, right? RNA maturation, export of mRNA from the nucleus. You'll get translation, modification with some kind of small molecule. Depending on the function, some things are modified, some things are not. Modifications are complete. That's a huge level of regulation in how the cells function. So not all proteins are modified the same. And then often you get degradation, right? When you need to turn something off, you'll degrade a protein that's crucial to the process. For example, with the cell cycle, right? We'll talk about that in depth. And when we talk about mitosis and DNA replication and all that stuff, right? Because that's part of the cell cycle. And remember, cyclins are required to interact with CDK, cyclin-dependent kinases, for that phase of the cell cycle to be on. When it's time to go to the next phase of the cell cycle, like exiting mitosis, let's say, you need to degrade all your mitosis-related cyclins so you can turn off those CDKs and move on into G1. So degradation is a very important. Who thought, and I'm not going to harp on degradation too much in this class because, I don't know, I just won't do that to you. But who thought that protein degradation in the ubiquitin proteasome system is all about putting misfolded proteins in the trash? Who thinks that? Right. It's not. It's about a lot more than that, right? It's about turning off, using degradation. Thank you for your honesty. It's about using degradation to turn off different cellular processes. That's the way the whole cell works. Increase gene expression, turn something on, decrease gene expression, degrade proteins, turn things off, all in response to some kind of stimuli from outside the cell. It sounds like people are ready to go, huh? I know, I got like 4 more minutes according to that clock. Who wants to... It's wrong? How come every semester that clock is off kilter? Okay, so you can go. Anyone, wait 1 minute. Anyone trying to get into the class who has a requirement for recitation, see me quickly.